Okay, so let's take a look at our active transport across the plasma membrane. So remember, active transport is going to require the use of ATP. So here, we need energy. Okay, so this is a little bit more complex of a reaction. So we're going to see that there's going to be 3 different types, and we're going to take a look at each one of these 3 methods. So the first is carrier-mediated active transport. Whew, that's a lot. So basically what we're going to see is in order for the molecule to move across this plasma membrane, it needs to have a protein with it. It basically needs to have the right key to get across the door. Okay? And it's going to need energy in order to do that. Usually what we're going to see is that the movement's going to be opposite to the concentration gradient. Okay? So let's take a look at the picture, because again, I think pictures help. And a great example of what we see in your body is this sodium-potassium pump across the cells. So here if we take a look, so right here, okay, on this first picture, we're intracellular means that we're inside the cell, extracellular means we're outside the cell. So here what we have is we've got material that needs to come across. In order for it to get across, right, you see that it's got to fit into the right thing, and it's going to have to require ATP. It needs energy to open the door, right? It needs energy to open the lock. So what's going to happen is that ATP is going to come in, the sodium comes in, and it allows it, because it fits the right shape, and the ATP is helping me know, right, helping that cell know, giving it energy to recognize the shape, it's going to allow it to move out. Okay. Now, obviously, that ATP is broken down into ADP, right, which remember we learned is going to end up getting recycled, right, over time. So this is important because, like I said, this is what allows the movement across the cell, but we need that energy in order to do it. The next methods are using endocytosis or exocytosis. Endocytosis is for material that's really large, way too big for the channel. Both of these, right? So we're not going to use, right here we use that protein channel. In endo and exocytosis, we're not going to use that channel. There's no hallway. So let's take a look at endocytosis first. What we're going to see is that the plasma membrane is going to engulf these particles, engulf particles in the cell and bring it out through their phagocytosis or pinocytosis. So let's take a look here. So basically what we have, all right, again, intracellular means we're inside the cell. Extracellular means we're outside the cell. So here we've got a particle that wants to come in the cell, but it's way too big, right? It's not going to fit across one of these tiny little channels. So what happens is you see it comes in and it kind of pushes its way in. What will happen is this little bulbous part will eventually close its way off, and you see it'll make a vesicle. And then, so this little vesicle, this phospholipid vesicle, is going to carry this particle to where it needs to go, right, in order to get checked out to move into the cell. And exocytosis is the opposite. This is removal. So endo is in, exo is out. Okay, so this is we're moving outside. So here we've got these vesicles that have lots of trash in it, right? And that trash is too big to go across one of those little tiny hallways of those proteins. So what's going to happen is literally just the opposite of we saw in endocytosis. So this little vesicle is going to come over here. It's going to attach itself to that phospholipid bilayer, the plasma membrane, and it's going to open up, and the contents are going to spill out. Okay. So this is going to help remove a lot of material from inside to outside the cell. All right, so that ends our active transport. So now we're going to take a look at the next section.